Hey guys, so we talked about diffusion in class today, and we're about to start our diffusion and osmosis lab, so I just wanted to give you a quick review of osmosis. We are in the textbook, chapter 5, page 106 and 108, if you want to go there and take notes on that and get more details than what I am providing you here. So remember when we talked about osmosis in class, we talked about diffusion in class. We were talking about traveling through a lipid bilayer, a semi-permeable membrane, and diffusion, remember we're talking about diffusion means no energy needed, and no energy is needed because it is the natural movement of the molecules, and here you can see there is a high concentration in the extracellular side of the cell, and so we will see these molecules moving to an area of lower concentration. And we can see that happening until it reaches, so I guess I should erase this because these are the steps here. So you see that we have the high concentration here. They begin moving across the membrane until they reach equilibrium. So molecules are always moving across the membrane, and this is called dynamic equilibrium. It never stops moving, but it's always trying to reach this dynamic equilibrium where the amount that is moving to one side equals the amount moving to the other side. When we're talking about osmosis, we're talking about the diffusion of water. And this is a little bit different than when we're talking about just straight up diffusion. Remember, diffusion by itself is talking about the movement of solutes. When we're talking about osmosis, we're talking about the diffusion of water. So if we look over here, these blue dots are water molecules, which this diagram kind of bothers me because it colors it blue, so it looks like there's something else over here besides just water, but these are the water. And when we're looking at a semi-permeable membrane, remember that not everything can cross that membrane. So in this case, these sucrose molecules are too big to cross. So the water is going to want to try and create equilibrium, and so it will cross to even out the concentration of solutes. So the water is going to move this way so that there's an equal amount of free water on both sides. And what do I mean by free water? Well, over here, there's a lot of free water because the water is by itself. And we know that when there is water and a solute, water, remember, attaches to the solute to dissolve it. So if this sucrose is dissolved in water, the water molecules around it are not free. So water in osmosis is moving from high free water to low free water. Or another way of saying this, it's moving from a low solute concentration to an area of high solute concentration. So the way that I remember osmosis is the water is going to move to want to be where there's more stuff, where there's more solute. So if you look at this picture over here of osmosis, this is kind of easier to visualize. We have the water over here, and if we're going to call this the solute in this case, this is solute, well, there's more free water over here because there's not as much solute dissolved, so there's more available free water. Well, we want to create an equilibrium. So the water is going to move to this side of this U tube to make the concentration of free water the same on both sides. And you can see here that the water level rises, and this is called osmotic pressure. So we can look at it both our ways. So again, let's make sure we can see it. So it's going from an area of high free water because there's less solute that needs to be dissolved in the water, to the side with more solute, aka less free water. So make sure that you can see both of these points in these two diagrams. So osmosis is a little bit different if we're talking about cells with cell walls or without cell walls. So right now we're talking about without cell walls. We are talking about animal cells here. So we know that the cell membrane is very liquid, it's very fluid. So if too much water comes in, it will pop. too much water leaves, it will shrivel up. And this all depends on the type of solution that the cell is sitting in. So here we have 3 examples. The first one is in a hypotonic solution. So hypo means below. So that means we have less solute in the solution compared to inside. We have higher solute inside of the cell, all the stuff in the cell. And what's going to happen, remember, is water wants to go to the high solute, so water is going to enter the cell. Well, if too much water enters the cell to create equilibrium, it's going to pop the cell, and that cell is now lysed. Remember, lysed means to break. In an isotonic solution, iso means equal, we have equal solute on both sides. So the solute on the outside equals the solute on the inside. Water is going to come in the same rate it goes out. This is normal. Our cells need to be in an isotonic solution. Or if we're looking at hypertonic, hyper means high. So that means that this cell is sitting in a concentration or in a solution with high solute compared to the inside of the cell, which is going to be lower solute compared to the outside. And in this case, since water wants to go to where there's high solute, it's going to leave the cell. And in that case, our cell is shriveling. So focus on that for a second. And a real life example of this is when you go to the hospital and you need an IV, there's different IVs depending on what your body needs. So if you need to replace body fluids, they're going to give you an isotonic IV solution. So they put saline. Saline is salt. So they're going to give you a little bit of salt water so that it equals the amount of salt on the inside of your cell, so that the water coming in equals the water going out, and it replaces the body fluids. If you need to make water leave your cell, so to reduce swelling here, they will give you a hypertonic IV, which means that it has more solute. And so if there's more solute in that IV they give you, and remember an IV is just pumping liquids into you, if there's more solute in that IV, then the water is going to want to leave your cell, and that will reduce swelling. If you need to rehydrate, so if you're dehydrated, they will give you a hypotonic IV solution. And what that's going to mean is hypo means less solute concentration in that IV. So when they pump it into your body and it is surrounding your cells, the water is going to go in to where there's a higher concentration of solute on the inside to rehydrate your cells. Osmosis is a little bit different when we're talking about cells with cell walls, because now if we have a cell walls are harder and they protect the plasma membrane, and they're not going to break, they're not going to pop. The cell membrane is not going to pop with the cell wall. And this is really important in plants because plants live outside and it rains, and they can't have, if it rained on these cells all the time, then your cells, or every time you got in the shower, you would have an issue with your cells popping. We don't want that in plants. So with the cell wall, it changes things a little bit. So in a hypertonic solution, so let's just say there is more solute on the outside, we know that water is going to leave. And what happens there is if too much water leaves, then the cell membrane detaches from the cell wall, and now your cell is plasmolyzed, shriveling up. If in an isotonic solution in plants, so that means equal solute concentration, the amount of water entering the cell and leaving the cell is the same, your cell is flaccid, meaning it's a little bit limp, because the pressure pushing up against the cell wall is actually what keeps the plant cells rigid and firm, and it allows the plants to stand up. So in a hypotonic solution, so again that means less solute in the solution, so there's more solute here, water is going to enter the cell, and what that's going to do, it's going to expand the cell, the inside of the cell is going to push up against that cell wall, and it's going to make your plants a little bit firmer. And this is good, and this is called turgor pressure. And that is the ideal situation for let's look at these two situations down below. This is celery, and this is an experiment that we can do in class. You can do it at home. If you put celery into a salt water solution, so if you're putting it in salt water, that is a hypertonic solution. And what happens? The cell is shriveling up. This is also an example because there's going to be salts on the inside. So it's still either it's isotonic or hypertonic. You're going to see the cells shrivel up. In distilled water, that means that we are in a hypotonic solution. There's less solute on the outside. There's no solute inside of distilled water. So when you put the celery in distilled water, the water is going to go into the plant cells. It's going to put pressure onto the cell walls, and now the celery, you can see, is firm and it's standing up straight. If you want to look at this under a microscope, you can see here this is an onion cell, normal. If we put the onion cell into 20% salt solution, so here we're talking about, this would be hypertonic. You can see under a microscope how now the cells are separating from the cell wall. So here's the cell wall. The inside of that cell is kind of shriveling up and detaching from that cell wall. If we put the onion cell into distilled water, so now we're talking about hypotonic, you can see that it returns back to the initial pretty normal looking onion cell because now there is pressure against that cell wall, that trigger pressure. So let's just do a couple of problems. So here we're talking about, these are problems with no cell wall. We will talk about problems with a cell wall later. But let's just see how we do with this. So if there is 70% solution on the inside, 30% on the outside, where is there more solute? It's on the inside, so water is going to want to go into the cell. And what's going to happen to that cell? It's going to burst. Let's look at B. So here we have 20% solution on the inside, 20% on the outside. That's good. That's equal on both sides. Water is equal on both sides. So we are in an isotonic, so it's going to go in and out. This is normal. And again, remember that vocabulary word. This would be isotonic solution. What would this be? We are in a hypotonic solution. But be careful with how you use these words, because if you're talking about the cell here, the cell in part A, cell is hypertonic to the solution, but the solution is hypotonic to the cell. So let's look at C. In C, we have 15 solute inside of the cell, 10% out. What's going to happen? Water is going to want to go in. And what's going to happen? The cell will burst. Let's look at it here. So on D, we have 15 in, 25 on the outside. What's going to happen? Think about it for a second. Water's going to want to leave. And what will happen? The cell will crenate. Crenate is another word for, shrivel. And this last problem. So I am on E. So this is something I want you to look at, is this 0.4, let me write it bigger, 0.4 M. M equals molarity. And molarity is just talking about a concentration. So we have 0.4 on the outside, on the inside, 0.6 on the outside. So there is more solute on the outside. So where's water going to go? It's going to go towards more solute. Let's see how we did out of the cell. And again, it's going to crenate or shrivel. So let's look at this last one. We have 0.3 on the inside, 0.4 on the outside. We're talking about concentrations. It's higher on the outside, so it's going to want to go